So hello students, so we are learning packed cell volume as an experiment in general hematology practical course. So here we'll know about what is packed cell volume. So let's know some basic background about packed cell volume test. This test is also known by hematocrit. Also we abbreviate or write some short form of hematocrit as ST or SCT. This test is also known by erythrocyte volume fraction, and we can define packed cell volume as PCV is the percentage of blood volume that is occupied by red blood cells. This is the definition of packed cell volume. That is what the percentage of blood volume which is occupied by red blood cells, that is the packed cell volume. होल ब्लड में रेड ब्लड सेल्स कितना है, तो रेड ब्लड सेल्स का परसेंटेज कितना है, दैट इज व्हाट वी विल लर्न अबाउट द packed cell volume. The value is expressed as a percentage or fraction of cells in blood. This is the value when we write the result of packed cell volume as a value. So we need to express as a percentage. Okay, the unit we have to write as a percentage or fraction of cells in blood, what we call it. Fraction of cells in blood means at what fractions the cells are present in the whole blood. Okay, for example, a PCV of 40% means that there are 40 milliliters of cells in 100 milliliters of blood. So out of 100 ml, 40 ml is a red blood cells. So this is what we define or express packed cell volume, right? There are two methods for performing packed cell volume test, PCV test. One is the macro method where we use Wintrobe's tube, and the second is the micro method where we use capillary tube, right? This is the requirement for macro method, okay, where we take Wintrobe tube and Pasteur pipette. You can see in the image there is a tube and there is a scale over there, 0 to 10 downwards and again 0 to 10 upward. So you can see the scale, and other requirements we have Pasteur pipette, okay, syringe, blood vials, stand we need, and the tube what we call Wintrobe tubes, and the pipette what we call as a Pasteur pipette, right? So these are the requirements we need to arrange so that we can perform the packed cell volume test. Macro methods using Wintrobe's tube method. So here, what are the properties of Wintrobe's hematocrit tube? The tube that we have. So how can we identify? How can we differentiate the Wintrobe's tube from another type of tube? So these are some characteristics. This is a special thick walled glass tube with a flat inner base. So inner base is a flat, okay, and the wall of this glass tube is very thick, and total length is 11 centimeter long. The diameter is 3 millimeter. The internal diameter is 3 millimeter, and it's calibrated at 1 millimeter interval to 105 or 110 millimeter. So this is the calibration which are done at the interval of 1 millimeter. It can hold about 1 ml of blood. So this is the characteristics of Wintrobe tubes. So we can put around 1 ml of blood, and this is total 11 centimeter long and calibrated at a 1 millimeter interval. Okay. Blood sample. This is the sample we need to collect it. Blood sample, and venous blood anticoagulated with the EDTA or heparin can be used. So this is the sample of choice. When we collect blood sample, so we need to take the anticoagulant, which one is a EDTA, or we can take heparin as anticoagulant. The process the sample without delay. This is what the instruction is there, that when we collect the blood sample, then we need to process the sample without any delay. If blood is kept at room temperature for more than 6 hours, swelling of erythrocytes can alter the test results. This is the sources of errors. We need to avoid it. and it is instructed that you need to perform the test as soon as possible after the sample collection. So without any delay, we need to process the sample, we need to proceed the procedure, and it has been observed that uh, more than 6 hours, after more than 6 hours at room temperature, what happens? The erythrocytes, which means red blood cells, usually undergo through the swelling process, and because of the swelling in the erythrocytes, it may can create some error in the results. So if a delay is expected, blood can be kept at 4 degrees Celsius for up to 24 hours. And now let's know about some procedure. What are the steps we need to do while performing this test? So first of all, mix blood bottle by repeated inversion or by a mechanical rotator. This is the instruction, the first step, that we need to invert. We need to do the inversion process, or we can put on the mechanical rotator so that the blood can mix properly with the the anticoagulant. And after that, once the blood is mixed properly, then we need to draw the blood into a long pasture pipette. So I have shown you the image in the previous slides. There is a long pasture pipette. Using that long pasture pipette, you need to draw the blood and fill the Wintrobe tubes up to 100 millimeter mark, starting from the bottom and gradually drawing the pipette as blood is expressed. So first of all, we need to you have to fill the bottom of the tube, and gradually you have to withdraw the pipette upward direction, so that you can avoid air bubbles which may can trap in the column of the the blood tube. Okay. After that, once you have filled the Wintrobe tubes, you have to centrifuge the tube at 2000 to 2300 gravity for 30 minutes. So this is the rotation given for centrifuge at 2000 to 2300 G for 30 minutes. You have to centrifuge. Here, to get this centrifugal force, you should get you have to set the speed at 3000 rpm in a centrifuge of 22.5 centimeter radius, and if you can set 3800 rpm in a centrifuge of 15 centimeter radius. So to achieve this this centrifugal force, this is the centrifuge has different radius. Okay, centrifuge has different radius. If you have chosen the centrifuge with the 22.5 centimeter radius, so you can set rpm 3000. If you have 15 centimeter radius of centrifuge, then you can set the rpm 3800. So this is what we can do it. We can centrifuge the blood which is present in the Wintrobe tubes at 3000 rpm for 30 minutes, or 3800 rpm for 30 minutes. Okay. Now interpretation. When we do the centrifugation process, now this is the interpretation you have to observe. The height of the column of the red cells is taken as a PCV. You can see the red cell. You can see in the image there is a black color, dark color spots are there. Okay, this is what it has shown. The color, the black color column of is red cells. Okay, and this is taken as a packed cell volume. The arrow indicate that the red cells are up to this level, and this height of the column of the red cell is considered as a packed cell volume. And what we understand here, that this is the volume occupied by red cells and expressed as a fraction of total volume of blood. So this is expressed as a total volume of the blood, right? Other fractions we can note are, we cannot Buffy coat layer. Okay, so Buffy coat layers can be seen where plasma can also be, we can see at the top, right? So what is Buffy coat layer? Buffy coat layer is a thin grayish red layer of leukocytes. So a gray layer of leukocytes or creamy layer of platelets above the packed red cells we can see, which can be just between the plasma and the packed red cells. Okay, so buffy coat layer is between the plasma and this packed red cells. The column of plasma is seen at the top uppermost. We can see the plasma is it's present at the top uppermost of the this tube. Now here we can present in different layers which can be seen in the Wintrobe tubes after centrifugation. On the first of the upper topmost is a plasma layer, and then after that there is a buffy coat which contain WBC and platelets, and now at the bottom we can see packed RBC, also known as hematocrit. So this is what we have learned in the procedure, and here we have explained what we have usually see after centrifugation. The normal range, we can see the normal range is in male, they have a adult male, they have a 40 to 54% of packed cell volume. In women they have 36 to 47% the packed cell volumes, and at birth the packed cell volume is around 44 to 62%. Now clinical significance, we can see this one. This is the clinical significance. Okay, a value below an individual's normal or below the reference range for the age and sex indicate anemia, and a high value indicates polycythemia. So based on the interpretation, based on the observation, what we do after procedure, when we get the result, so we can be able to discriminate two conditions. We can see if there is anemia, or we can see if there is a polycythemia. What is anemia? Anemia is a condition when packed cell volume is less than normal range. Okay, if you are doing packed cell volume test, so based on packed cell volume test, we can be able to diagnose anemia like condition. So at what condition we can report the anemia? When the value is less than normal range, and if the value is more than upper limit of the normal range, then it can be considered as a polycythemia. Okay, polycythemia is a condition when packed cell volume is increased above the normal range. Hematocrit reflects the ratio of red blood cells to plasma and not the total red cell mass. So this, you have to understand that the hematocrit, what we call as a packed cell volume, it is a ratio of red blood cells to the plasma. That in the whole blood, how much is the red cell and how much is the plasma. Okay. For example, hematocrit may be below, may be low in pregnancy, as there is a hydremia, because in the pregnancy the blood volume is increased, and we call them as a hydremia or hemodilution. Okay, so there is a hemodilution, the water content in the blood is increased, so at that moment hematocrit becomes lower side, and here the total number of red cell mass is not reduced actually, but the plasma volume is increased. So because of that, what we say that the hematocrit reflects the ratio of red cells to the plasma only. Okay. The opposite is seen in the hemoconcentration. What happens in hemoconcentration? The red cells remain, but the plasma volume actually decreases. This is the comparison between the Wintrobe tubes method and the capillary tube method. This is the Wintrobe tubes method is a macro method, and capillary tube method is a micro method. Okay, and if you compare this Wintrobe tubes with the capillary tube, so the first one is the length of the Wintrobe tubes is 110 millimeter, whereas capillary tube the length is 75 millimeter. And second, the diameter of the Wintrobe tube is 3 millimeter, whereas diameter of capillary tube is only 1 millimeter. Wintrobe tubes, we can see it is graduated from 0 to 100 millimeter, but capillary tube is not graduated here. And the sample you require for Wintrobe tubes is a EDTA or oxalated or heparin. Okay, but here capillary blood, we can use capillary tube. Okay, heparinized capillary tube, or you can also use plain capillary if EDTA or oxalated blood is used. So here sample can be taken similar for both capillary tube and Wintrobe tubes. Now the centrifugation at 3000 RPM for 30 minutes is required when we do the procedure. That the Wintrobe tubes with the blood should be centrifuged at 3000 RPM for 30 minutes. Whereas we can see that in this capillary tube method, centrifugation at 15,000 rpm is required for 5 minutes. So you can see the difference in the centrifugation process, and the scale with the descending order is used for PCV determination. Like you have seen the picture of the Wintrobe tube. There are two scales. One is labeled as a 0 to 10 upside direction, another is labeled as a 0 to 10 downside direction. So the scale with the descending order is used for PCV determination. Okay, descending order means what we see that from uh, from downward to upward direction. Okay, we can just take the collect the packed cells, whatever the level of packed cells are there, and after that we can give the results. And here the reading is noted by holding the tube against the hematocrit scale. So there are hematocrit scales are there, and after centrifugation we can hold the tube against the scale, and the bottom of the red cell is aligned with the horizontal zero line. So here the horizontal lines are there in the scale. So bottom of the tube is aligned to the horizontal line, and we can be able to measure the level of packed cells against the hematocrit cells. So we have learned here today. We have learned the packed cell volume with the by Wintrobe methods, and we have compared this method with the capillary tube method. Okay, though this capillary tube methods is not used these days because here a special type of centrifuge is required, and also the centrifugation rpm is 15,000 rpm. So this is the a special centrifuge, as well as the high speed centrifugation is required, and even if we require hematocrit scale also this separately. So we do not use capillary tube method these days, but Wintrobe's method can be used in the lab. And but now also this Wintrobe method is also has gone outdated, because we get this results with the help of analyzer, which is available in the even in the medium class lab also. Okay, so but when the analyzer is not working at the moment, we can perform the PCB by manual method, the Wintrobe method. Now we have completed. We have come to the end of the slide. Now, if you have any questions, we are open for discussion now. Yes, any questions?